For the Mannitol salt agar test, there are a couple of materials that you will need to gather. These include your Mannitol salt agar. I don't actually have it, so we're just going to pretend that it is in this test tube right here. Our unknown bacteria stock culture, which is right here. An agar plate, our inoculating loop, and our Bacto incinerator. To start the experiment, first you're going to sterilize your inoculating loop in the Bacto incinerator for about 5 to 10 seconds. Once you've done that, allow it to cool off. Once the loop is cooled, you're going to obtain a little bit of your unknown bacteria from your stock culture using your sterilized inoculating loop. You're just going to scrape some of that out. The cap back up. After you've allowed your inoculating loop to cool off, you're going to obtain a little bit of your stock culture of your unknown bacteria using your sterilized inoculating loop. So you're going to take the cap off, flame the test tube for 5 seconds, then take your inoculating loop and scrape out a small bit of the culture. Flame the test tube again for 5 seconds. Then put the cap back on. Put it back on the test tube rack. And now what you're going to do is you're going to take your agar plate and you're going to do the streak method using your inoculating loop that contains your stock culture. What you're going to do is just spread the culture like so. Kind of like this. You want to cover the entire plate with it, going from left to right. Put the lid back on, sterilize your inoculating loop, then set it back on your bench. And now, after you've done all that, what you're going to do is you're going to take your Mannitol salt agar, pretend that I have some of it in here, take the cap off, and you're going to pour this into your agar plate, like so. Put the lid back on, put the cap back on to the test tube, put it back on the test tube rack, and then let your culture incubate. After your culture has incubated and you've observed and analyzed your agar that contained the culture, what you're going to do is you're going to get rid of that culture by putting it in the biohazardous waste container, which I have already done. And then you are going to decontaminate your workspace by washing it down with germicide and wiping it up / drying it off with paper towels. Make sure to dispose of the used paper towels in the biohazardous waste container. After that, you can disinfect yourself. First, remove your gloves and put them in the biohazardous waste container as well, and then wash your hands thoroughly. After you've done that, go and grab a new pair of gloves because you will be conducting the next experiment, which involves using a starch agar.